This is section 17-1, Genes and Variation. So this is the second chapter for our unit on evolution. So how is evolution defined in genetic terms? So we've been looking at evolution and how the theory came about and what the evidence was for evolution. Now we're going to look at it, how it relates to genetics. So in genetic terms, evolution is a change in the relative frequency of alleles. So how often are those alleles present in a certain population? You have what's called the gene pool, and the gene pool is all the alleles that are present in a population for a certain gene. Okay, so if you remember when in Darwin's time, Mendel hadn't published his information yet, so Darwin didn't know that heritable traits were controlled by genes. Okay, and he did not know that genes, which are on chromosomes, are what generate variation. So let's review some of the terms from genetics. You should remember that a genotype is all the alleles for a gene or that a person has. Phenotype is going to be the physical, or how the person looks, or what traits the person has. Don't forget that some of those traits could be physiological. For instance, maybe you're missing an enzyme to digest milk. Okay, so that would be a physiological phenotype or a trait. Also, your behavioral characteristics, how you walk, how you talk, those are all controlled by your genes. Also remember that the genotype is produced, the genotype along with the environment gives you your phenotype. So for instance, you may have the alleles for melanin, which makes your skin darker, but your skin won't be darker unless you go in an environment where there's a lot of sun. Okay? So natural selection acts on phenotypes, not genotypes. Natural selection acts on phenotypes. So for instance, down here, if you look at the picture I have here with the crows, they eat the green beetles. Do they eat them because they're green? Or do they eat them because they have the green genotype? No, they eat them because they look green, not because they're big G, or big G, little g. Now, as far as a population, you should remember that a population is a group of individuals of the same species that can mate and produce offspring. Okay? And their gene pool would be all the genes, including all the different alleles that are there for that population. Okay, so each organism may have different alleles, but the gene pool would be all the alleles present for the whole population. So when we're looking at relative frequency, we're looking at the number of times that a certain allele occurs in the gene pool. So, for instance, if you look at the beetles on the left, the first generation here, my pen working. Okay, the first generation here, we have 6 total beetles, right? How many alleles do we have present in this first generation? Well, we have 12 alleles. Okay? How many green alleles do we have? We have 9 green. I'm going to put a little g for green because green is recessive. We have 9, right? And then for the big G, which would be our brown one. I guess I should have used brown, but okay, I'll do that. Let me erase this. Okay, so brown is dominant over green because in the heterozygous condition, brown is dominant. Okay, so how many brown alleles do we have? We have 3. And the green alleles, we have 9. So if you try to figure out the frequency of these, for big B, you would have what? That's right, 25%. And for little b, you would have what? 75%. So that's how they figured out the 75 and 25 here. But if you're looking at the phenotype, we have what? It's 50-50, isn't it? Green ones to brown ones. Okay? Now this diagram shows the fur color in a population of mice. So let's look at how many black mice we have and how many brown. So just say we have 100. That makes it easy because you have 48%, 16%, and 36, and that's going to equal 100. So on the, if we're looking at the number of alleles, we have black alleles, right? We have have black alleles, I'll put a big B, and then we have brown alleles, which is little b. So how many little b alleles do we have? If there's 36 mice and they all have 2 alleles, that would be 72. 36 mice times 2 alleles each would be 72. Now for the homozygous black, there's 16 mice. How many alleles would that be? That's right, there'd be 32. Now on these heterozygous ones, they have an even number of black and brown alleles, don't they? So if there's 48 mice, they would have 48 brown and 48 black alleles, wouldn't they? Okay, so that gives us a total of, let's see, 80, does that sound right? 80, and then this one equals 120. Now as far as a percentage, for the black allele, you divide by 200, because the total number of alleles we have is 200, right? And that would equal 40%. 40, okay, that would equal 40%. And then this one's 120, and that would equal 60%. So if you look, if you're just doing black alleles to brown alleles, it's 40-60. And then as far as the actual genotypes, the ratio was 48:16:36. So let's review our definition of evolution in genetic terms again. Evolution is the change in the frequency of alleles in the gene pool of a population over time. So before, when I gave you a definition of evolution, we just said what? We said change over time. Now when we add genetics to it, we have to look at the frequency of alleles in the gene pool. So we'll always look at the percentages to see if those percentages of alleles are changing. Okay? And we also know that natural selection operates on individuals. Individuals get selected, not whole populations. And the population will evolve as opposed to an individual. Individuals' alleles are not going to change, but a population's gene pool of alleles will change. So what are the sources of genetic variation? Well, there's 3 sources. You have mutations, you have genetic recombination, and you have lateral gene transfer. And I'm going to talk about each one of these. So for instance, mutations are changes in the phenotype that may or may not affect fitness. Now on a smaller level, it's actually mutations on the chromosome or of the genes. And they can be bad or they can be good. So they can increase fitness or they may not increase fitness. Now these mutations have to occur in germ-line cells. You remember stem cells, I'm sure. Germ-line cells are the ones that become your gametes. Okay? Germ-line cells become your gametes. Okay? Now, as far as recombination, do you guys remember independent assortment and crossing over? If not, you probably should look those up. But crossing over this is what this picture is here, is crossing over. And then independent assortment basically was saying that traits will assort independently, or the genes assort independently, so you can have different combinations. So you could have brown eyes with brown hair, brown eyes with red hair, brown eyes with black hair, just as you could have green eyes with brown hair, green eyes with red hair, green eyes with black hair. That's what that's talking about when it says independent assortment. Lateral gene transfer, do you remember Griffith? Okay, and Griffith did the experiment with the mice. Okay, and remember the mice, the bacteria was harmless, and then he mixed it with the dead bacteria that was harmful, and then the harmless bacteria became harmful. Well, that would be an example of lateral gene transfer. So when we're looking at phenotypes, what will determine how many phenotypes a gene will have or a trait will have? Well, it's the genes. It's the number of genes that control the trait. So if it's just one gene that controls the trait, you might have 2 or 3 different phenotypes, maybe even 4. But if it's a couple of genes, or if it's polygenic, you could have a numerous amount of phenotypes. So a single gene trait means that it's controlled by one gene, and it could have 2, 3, or even 4 in the case of blood types. So if you think of blood types, you have, it's the I allele, or I gene for immunoglobulin. That's why we wrote it like this. Remember, IA, we had IB, and then we had little I. It's still the I gene, it's just a matter of which markers are on it, which antigens were on it. Now, obviously, if a trait or an allele is dominant, that does not mean that it's dominant in the population. And when I say dominant in the population, I mean greater frequency in the population. Okay, because sometimes a dominant allele or a dominant trait, or the dominant form of a trait, is not always good. Okay, so for instance, Huntington's is a dominant disorder, and a lot of people, most people don't have that. Polygenic traits are controlled by 2 or more genes, and they can have several different phenotypes. An example of a polygenic trait would be, here we have height, and it's what's called a bell-shaped curve, and so all the different heights from the shortest to the tallest are all different phenotypes. Okay. On the review questions, I believe you should be able to answer these. Okay. And that brings us to the end of this one.